Scientists say they have used the gene editing tool CRISPR inside a human body for the very first time. The treatment is a new step in efforts to treat diseases by operating on DNA, the chemical code of life. CRISPR stands for clusters of regularly interspaced short palindromic repeats. CRISPR technology is a powerful new tool for editing DNA, as it allows scientists to alter the DNA and therefore the function of genes. A patient recently had their DNA edited using CRISPR at the Casey Eye Institute at Oregon Health and Science University in Portland, Oregon, USA. This is the first time this treatment has been done inside a body. On March 4th, the companies that make the treatment announced that the treatment was for an inherited form of blindness. It may take up to a month to see if the treatment to restore vision was successful. And if all goes well, doctors plan to test it on 18 other patients. We literally have the potential to take people who are essentially blind and make them see, said Charles Albright. He is the Chief Scientific Officer at Editas Medicine, the Cambridge, Massachusetts-based company developing the treatment with Allergan, a Dublin company based in Ireland. We think it could open up a whole new set of medicines to go in and change DNA. Jason Commander is an eye surgeon at Massachusetts Eye and Ear in Boston, another hospital that plans to enroll patients in the study. He said, "The surgery makes a new era in medicine, using a technology that makes editing DNA much easier and much more effective." Doctors first tried in-the-body gene editing in 2017 for a different inherited disease using a tool called zinc fingers. However, many scientists believe CRISPR is a much easier tool for locating and cutting DNA at a specific spot, and interest in this new research is very high. The people in this study have a disease known as Leber congenital amaurosis. The condition is caused by a gene mutation which stops the body from making a protein that converts light into signals for the brain to process, thereby enabling sight. People with the condition are often born with little vision and can become blind after a few years. Scientists cannot treat this condition with standard gene therapy. Normally, gene therapy replaces a defective gene, but with this medical condition, the gene that is needed to treat the disease is too big to fit inside a disabled virus that helps transport the replacement gene into cells. Instead, scientists are trying to edit or delete the mutation by making two cuts on either side of it. The hope is that the ends of the DNA will reconnect and allow the gene to work as it should. The treatment is done in an hour-long surgery under general anesthesia. Through a tube the width of a hair, doctors drip 3 drops of fluid containing the gene editing machinery just beneath the retina. The retina is the lining at the back of the eye that contains the light-sensing cells. Once the cell is edited, it's permanent, and that cell will persist, hopefully, for the rest of the patient's life, said Eric Pierce at Massachusetts Eye and Ear Hospital. Pierce is a scientist who was not involved in this first case. Doctors think they need to fix 1/10 to 1/3 of the cells to restore vision. In animal tests, scientists were able to connect half of the cells with the treatment, Albright said. The eye surgery itself poses little risk, doctors say. Infections and bleedings are relatively rare complications. One of the biggest possible risks from gene editing is that CRISPR could make accidental changes in other genes. The companies have worked to minimize this risk and to ensure that the treatment cuts only where it is intended, Pierce said. The gene editing approach is really exciting. We need technology that will be able to deal with problems like these large genes, said Jeanne Bennett. She is a University of Pennsylvania researcher who helped test Luxturna at the Children's Hospital of Philadelphia in Pennsylvania. Kiran Musunuru is another gene editing expert at the University of Pennsylvania. He said the treatment seems likely to work based on tests in human tissue, mice, and monkeys. The gene editing tool stays in the eye and does not travel to other parts of the body. Therefore, if something goes wrong, the chance of harm is very small, he said. It makes for a good first step for doing gene editing in the body. The new study is the first to use CRISPR to edit a gene inside the body. Another company, Sangamo Therapeutics, has been testing zinc finger gene editing to treat metabolic diseases. Other scientists are using CRISPR to edit cells outside the body. They are trying to treat cancer, sickle cell, also known as red blood cell disorders, and some other diseases.